welcome to R.K Medicos. In this section, we are going to discuss about the general embryology part 4. In general embryology part 4, we are discussing about the third week of the development of human embryo, and also we are going to discuss what are the main processes is occurring in this third week. So let's start our discussion. In the third week, we are also seeing there is a placental development here, and this placental development, how they are going to develop from the primary, secondary or the tertiary villi, they are going to invade at different parts. And in this third week, we are also going to discuss about the two main process, that is gastrulation and noodulation. What does it mean, gastrulation here? Basically, the gastrulation, it means simple, the formation of germ layers. And what is germ layers? Do you know from the third part? What we discuss in the third part, the germ layers are the ectoderm, mesoderm and endoderm. And one more in the recent update, that is the neural crest cells. So these are the 4 germ layers for now, and there gets updated version, that is neural crest cells. So ectoderm, mesoderm, endoderm and neural crest cells, they are the 4 simple types of the germ layer. They all germ layers are forming from the either hypoblast cells or epiblast cells. Where these cells are formed, epiblast mainly, these cells are forming them, we are discussing here. Next process is noodulation. Simple, in the noodulation we are going to see the formation of neural tube, and from this neural tube in the later stages there is a forming our cranial part or the central nervous system part. These all the nervous system part is developing from them, that is spinal cord or the either brain, these all are forming from the neural tube. So this process begins also in the third week of the human development. So now you tell me the two processes, which one? That is gastrulation and noodulation. Further we are moving here. When these processes are occurs, these processes are occur in third week. Simple. And the formation of germ layer, formation of neural tube. These are the two processes we have to discuss in this whole third week development and also placental development. So let's start our discussion about the gastrulation. Basically, and gastrulation means simple, the formation of germ layers, that is ectoderm, mesoderm and endoderm. Let's say from the initials, you know some points here, these upper cells and the lower cells. The upper cells are going to in columnar in shape. They are columnar, the upper cells. You can see here, these are the columnar, and these are called as epiblast cells. Epiblast cells. Same here we can see in the down there is a cuboidal cells, and these cuboidal cells are called as the hypoblast cells. This is happening in the second week. Now we are moving towards the third week, and what happened in the third week with these cells? What the cells? These cells are also called as embryoblast cells. They are going to form our embryo. These are the inner cell mass, and the outer cell mass forming the trophoblast layer. These are providing nutrition. In previous section we already discussed cytotrophoblast, cytotrophoblast. These are the conversion of the trophoblast layer. And here in the inner cell mass there are the two parts, the epiblast and hypoblast, and that is embryoblast cells. The epiblast cells is in third week going to proliferate and form something. This epiblast, the upper part cells, is going to modify here, and they forming a simple asterisk like a structure. This asterisk is formed first time, that's why they are called as primitive asterisk. So now you tell me the primitive asterisk is formed on which week? That is third week. From which cells? Epiblast cells. Same, the hypoblast cells is going to proliferate in the columnar cells. These hypoblast cells are going to proliferate and converted their cuboidal cells. Their cube-like cells is converted into cuboidal. So this is the modification, and these are called as prochordal plate. Prochordal plate. So now you tell me there is two terms, that is the primitive asterisk and prechordal plate or prechordal plate. These are developing from the epiblast and hypoblast part. Now this primitive asterisk is forming something, and the prochordal plate is forming also something. The prochordal plate is going to develop further in the buccopharyngeal membrane, basically bucco in the buccal region or the mouth region, and in the pharynx region. These structures are basically formed by which part? That is hypoblast part, and the prechordal plate is further going to develop the buccopharyngeal membrane, and after this suppose that this is the buccopharyngeal membrane, and this membrane is going to rupture here. The rupturing of this membrane is opening of our mouth. Simple. So this rupturing of membrane is opening up our mouth, and mouth is situated towards the cranial end in our body. When suppose that this is any person here, this structure is called as cranial part, and this structure is called as caudal part. Simple, caudal. So this is cranial, this whole structure is caudal. So now you tell me, the mouth is situated in which part? That is of course in the cranial part. So the precaudal plate is going to develop our head end, or we can also say cranial end. Same, primitive streak is going to form anal region. That's why they are called as caudal end. Simple. This structure we are going to revise in the below picture. Let's look on this gastrulation process. And I also told you, you can see here, these modifications of hypoblast cells are called as precaudal plate. Simple. And they are also called as head end, because they are going to rupture of the buccopharyngeal membrane is forming by this precaudal plate is rupturing, and they're forming the mouth oral cavities. Same, there is epiblast cells, columnar cells are going to modify it, and they're forming the primitive streak. You can see here, this primitive streak is formed by these epiblast cells, and the proliferation from the epiblast cells, this is called as the gastrulation begin. Now you tell me, what is the gastrulation begins means here? In the third week, the gastrulation is started. Why? In the third week, the epiblast cells are going to proliferate. Why? The proliferation of these epiblast cells is forming something that is called a primitive streak, and later on this primitive streak structure is going to develop in the different parts, ectoderm, mesoderm and endoderm. So we are going to learn here. And this primitive streak part is also called as tail end. So now you tell me, tail end is present in which part? Primitive streak. Precaudal plate is, that is at the head end. Now you can see here, there is some terms, dorsal and ventral. What does it mean? The dorsal and ventral term. These are present in the ventral part of our embryo, and this is in the dorsal part. So now you tell me, the precaudal plate is present in the ventral part, and the dorsal part is what? In the primitive streak. So we can see the germ layer formation is in the craniocaudal sequence. We can see here, suppose that any person here, I already told you this one is cranial part, and these all parts are caudal part. So the first what structures is formed in the embryo, that is our cranial part. This is the first formed structure, and later on what structure is formed? Secondly, they are forming caudal. So this sequence of formation of our embryo, in the what structures are formed, that one is called as craniocaudal sequence. Simple. And these germ layers are forming craniocaudal sequence. How it's going to develop here, we are going to see. First point, first we revise it. The craniocaudal sequence, the germ layers are going to proliferate. The hypoblast cells are the cuboidal, the epiblast cells are the columnar, the precaudal plate are the columnar cells of the hypoblast. They are converting from the cuboidal. They are the cells like a cube-like. These cube-like cells are going to proliferate into columnar, and they are forming prochordal plate. So now you know these terms. Let's move on. Next, say, we can see here, I already told you this one also, the precaudal plate or the prochordal plate is forming something, that one is called as buccopharyngeal membrane, and this buccopharyngeal membrane, suppose that this is going to rupture here, and the opening of our oral cavity at the mouth. So the rupturing of buccopharyngeal membrane is the developing of our oral cavity. Simple. Next we can say this prochordal plate, what the prochordal plate is developing from the hypoblast cells is forming the head end of the embryo. It is true, I already told you. When the primitive streak is called as, why the primitive streak is called as tail end, already know it. These are developed from the epiblast cells, and the epiblast cells are participated in the formation of the caudal end. So they are called as tail end, and they are derived from the epiblast cells, you can see. And the formation start from the end of the second week and the beginning of third week. They may be start from the 14th day of the development of embryo. So we can also say on the 14th day, it means second week. It means they are starting from the either 14th or the 15th day. So we can say like this, that at the end of second week or the beginning of third week. So indicate the starting of gastrulation. Already told you, when there is a beginning of formation of primitive streak from the epiblast cells, is the marking of, is the sign of the gastrulation which begins in the embryo. So we can see what happened on the day 15th. Primitive streak is formed. Yeah, we can see there is a two terms. Suppose that this is the embryo here. Embryo. What happened to this embryo? We can see there is a two structures are developed from the epiblast and hypoblast cells. What the structure developed from the epiblast cells, that is here. And what the structure developed from the hypoblast cells, that is here. Now you tell me. Hypoblast cell structure developing prochordal plate. They are going to form our oral cavity. They are the head end. They are the cranial end. It is simple. Same. The tail end is going to develop our primitive streak. Simple. And they are forming from the epiblast cells, the upper one. And these epiblast cells are going to develop primitive streak. Here is the another structure, that is cloacal membrane. These all structures are developed in the third week, beginning of the third week. So now we are moving further. What happened to these patients and why we are looking on this? First, you know from our first part of the general physiology, general embryology, we already discussed about it. The sacrococcygeal teratoma. Teratoma means simple, tumor of germ layers. And I already told you, from the germ layers, a whole body is going to form, or the whole body parts of any organism is going to develop from them, and there is any tumor in that region. The primordial germ cells are moving somewhere, and these primordial germ cells are going to modified, or their version is going to suppose that there is a tumor of that cells, it leads to the sacrococcygeal trauma in the sacrococcyx region. So we see that time the two regions of it. The one is the abnormal migration of primordial germ cells, you know from the first part. Now here second part, the primitive streak persistence. The persistence of primitive streak is also leads to the what? The development of sacrococcygeal teratoma. Why? We can see here. Here is the primitive streak, and primitive streak is going to present in which region? Tail region. Simple. When they are going to proliferate, because the tumor, it means simple, the abnormal division of cells. And in that region, in the primitive streak, when there is abnormal unregulated control of these cells, or the revision of these cells, it leads to the development of teratoma in the sacrococcygeal region, because they are present in the tail region. Simple. So when you know that part of first part of embryology, it's very simple for you why it's happened and what is going on here. Next we can see here. Next, move on the next topic. There is a germ layers development. How the germ cells or the germ layers are gets formed in the different parts of the our embryoblast cells. We can see here the germ layers development in the first germ layer. Which is the development of which germ layer is going to develop first? First we can see, we know the craniocaudal sequence. The first cranial parts gets formed, and then caudal part is going to develop. All I told you is according to structure. And the first part is developed from the first part, or the first germ layer is developed in any embryo. The first germ layer is developed, that is the endoderm only. Endoderm. Now you tell me, the endoderm is the first germ layer development? Yes. And they are developed from the which cells? Epiblast cells. So the epiblast cells are first going to modified into the primitive streak. They are further going to forming a germ layer. That one is called as the endoderm. Simple. Next we can see the second germ layer is going to develop is the intra-embryonic mesoderm. Simple. And the third one is developing is called as ectoderm. Already told you the updated version is having 4. So the fourth one is called as neural crest cells, or they are also called as NCC. So these are the 4 different types of germ layers are going to develop here. And now you know the first germ layers are going to develop from the epiblast cells, the endoderm. And now you can imagine here this endoderm germ layer is going to replace the hypoblast cells. So first we can see here, this is the primitive streak. and from this primitive, and this is epiblast cells. Basically this is not epiblast, this is the lining epiblast, this region, and these are the amyloblast cells. Amyloblast cells, these are the covering of which cavity? Amniotic cavity. Here amniotic cavity structure we already discussed in the second part or third part, amniotic cavity. And below which cavity is here? Which is the cavity located here? That is yolk sac. And there is the epiblast here. Wait a minute. This is epiblast and this region is hypoblast. You don't know? Don't worry about it. I'm going to erase it and we are come again on this. So leave it for now. Now above the epiblast there is a amniotic, below it's yolk sac. And primitive stick is the structure developing in the epiblast or hypoblast. Of course epiblast, already discussed. So here you can see these are the cells. These are the cells are forming the primitive stick. Simple. These are the cells we can see here. These are forming the primitive stick. Further these cells are going to move down, and they are forming which cells? Now you tell me. They are going to form first germ layer, that is endoderm. And we can see this is the endoderm here. And these endoderm are moving further down and they're going to replace. These are the hypoblast cells. These hypoblast cells are going to replace here. These hypoblast cells are going to be replaced by which cells? These hypoblast cells are going to be replaced by the endoderm cells. So the endoderm cells are forming from the primitive streak, and after that they're coming down and they are going to moving these hypoblast layer cells aside. Simple. So this is the concept of the formation of the second part, that is the first germ layer of the that is the endoderm. And we can see next we are going to see the second one. The second germ layer is derived from the primitive streak, and that is the intra-embryonic mesoderm. And these are having two structures in the second germ layers of development, that is the primitive streak. They are derived from the primitive streak, and that is what is the structure derived, that is intra-embryonic mesoderm. And you now know the primitive streak is same. They are developed from the epiblast or hypoblast. Of course epiblast. The intra-embryonic mesoderm is present or spread throughout the disc. This whole structure, the epiblast or hypoblast, is called as bilaminar disc. I told you these are the two layers of the cells, that is hypoblast or epiblast, and they are present in a same linear manner. That's why they are called as bilaminar. In the lamina they are present in a laminar manner, and that's why they are called as bilaminar disc. So inside this disc there is the intra-embryonic mesoderm is going to spread. So that's why they are called as, they are spreading throughout the disc except somewhere where these are not present. So where these structures are not present, these are not present in the prochordal plate and the cloacal membrane. In these two regions, the cloacal membrane or the prochordal plate, there is a absence of what? Intra-embryonic mesoderm. Either they are spreading throughout the disc, or what is the region we are going to see here on the picture. Maybe it's clear for you here. You can watch on it. Now we already see these are the what cells? Endoderm. And these endoderm cells are replacing the hypoblast cells. Now we are going to move on the red cells. Look on the red. I'm going to mark on the yellow. So you can see these are spreading in all this region. We can look here. Same here too. These yellow cells are present all over the disc. How the disc is formed? This is the disc here. You can also see this is the disc here, and this is the upper part of disc, and this is the lower part of disc. The upper part of disc is made by epiblast, and epiblast is forming primitive streak. Simple. The lower part is formed by hypoblast, and inside this disc, most of the cells of the endodermal origin, they are the intraembryonic mesoderm are present. They are going to modify it from the cell. Same we can see here. This is the primitive streak. This is the cells of primitive streak. Suppose that this is the primitive streak, and they are going to form both. They are going to form either these cells, and they are going to form either these cells. And these are the endoderm cells, and these are the intraembryonic mesoderm cells. So these both the cells group are formed by the primitive streak, and primitive streak is formed by the epiblast cells. So it's simple. The epiblast cells, or the either endoderm, or they are developed from the intraembryonic mesoderm, is also developed from the primitive streak. It means both are formed from the same, that is epiblast cells. So hope it will be clear for you now which are the cells going to form the endoderm or the intraembryonic mesoderm. And now later on we are going to discuss this picture. For now we moving towards the third structure, and now you know what are the structures. They are not going to cover it. That is the prochordal plate, that is the prochordal plate and cloacal membrane. In this region there is no intraembryonic mesoderm is present. In this picture we can't see, wait for it, we look on the another picture and you can imagine it. Made only by the, now you imagine if any structure is not covered by something or not made by something, it means they are made by something else. And now you know there are three germ layers mostly, that is three germ layers, that is ectoderm, mesoderm and endoderm. Now you know endoderm, mesoderm, ectoderm. Now you told me when the any structure is not forming from the mesoderm, it means so simple, it is forming from the another two structure. So the cloacal membrane and the prochordal plate, these are the two structures are developing from that. They are covering through the what structures, that is the ectoderm and endoderm. They are not going to form by the mesoderm because mesoderm is not present in that area. Already told you. So it's so simple. Now we are moving towards the third germ layer, that is ectoderm. And the ectoderm, what the structure is remains, the remaining epiblast cells, what the epiblast cells is remain in that area. These all the epiblast cells are going to develop and they forming the intraembryonic mesoderm after the formation of intraembryonic mesoderm in the disc manner. What the cells present in the epiblast, the left cells, these cells are going to modify and form ectoderm. Simple. So the ectoderm is formed by which cells? Once again they are the epiblast cells. So all the three germ layers are developed from the epiblast cells. No, this is the golden line here. All the germ layers are developing from the epiblast cells only, except something. All the germ layers are derived from the epiblast, and we can also select this more than yolk sac. Yolk sac is the cavity just below the what? That is the hypoblast cells. Now why we are called this yolk sac is greater than epiblast? In early books the yolk sac is mentioned there, they are formed by the yolk sac. But for now the new concept, the epiblast cells are developing everything. And now you know everything how they are going to develop. So the first primitive stick is formed. After that the cells going to divide in two manner, that is in the middle. The first germ layer is formed, that is endoderm. The cells are coming and they replaces the cells of the hypoblast and they are forming the endoderm layer. After that in the second germ layer formation, that is intraembryonic mesoderm, they are going to form and they are moving in the more whole disc manner in the between the hypoblast and epiblast cells. After that we can see the third germ layer, that is the endoderm. What the remaining cells of the epiblast is going to divide and forming? Simple, that is endo or ecto, that is ectoderm. So now we are moving towards the fourth germ layer, that is the neural crest cells is developing from the secondary mesenchyme. We discuss later what is the secondary mesenchyme or primary mesenchyme. So for now it's all in this topic. Next we are moving towards the notochordal development. In the notochord development, all I told you, in the notochord development we can see here in this picture. Once again, this is the embryo. And all I told you, in the embryo two parts are formed, that is epiblast and hypoblast cells are going to develop in the precaudal plate. Is what is the development by the precaudal plate is from what the cells they gets developed, that is the hypoblast or epiblast. Hypoblast. And primitive streak is formed by the epiblast. And this primitive streak down to its in the caudal, they are also called as caudal end. Caudal end, the structure is developed in the caudal part of the primitive streak, that is called as cloacal membrane, and on the primitive streak cranial end towards the, this one is the cranial end, and here is the caudal end of what? Of the primitive streak. And this one is primitive streak whole structure. Now you imagine the primitive streak is having two parts. We are saying like this is the cranial part and this is the caudal part. The cranial part is going to thickening. They are going to thicken. It means the primitive streak, what the part of the primitive streak is going to thicken in the cranial end, and they forming a node. That node is called as primitive node. So the primitive node is formed by the thickening of which part? That is primitive streak, and it is developing from the all cells by the epiblast cells. Now you can see in this picture also same is happening here. This is the embryo here, the whole structure. Wait a minute. This is the embryo, and the whole embryo, inside the embryo there is a head end and tail end. Head end is basically prochordal plate, that look on this, and the primitive streak is the whole, this structure is the primitive streak. And below the primitive streak, now you know, already told you, here is the cloacal membrane, and they are going to develop in the different regions, see later. But for now, primitive streak is going to thicken here. Look on this. Primitive streak is going to thicken here, and they are developing something in that region, a node. That one is called as primitive node, and that another name of it is called as Hansons Node. Hansons Node. And they are developing a peat-like structure here around peat. That's why they are called as the primitive peat. So now you told me the thickening of, you can also see here the thickening of primitive streak in the cranial end. Simple. So now you tell me the primitive peat, Hansons node, and the primitive streak all are derived from the epiblast cell. Simple. It's so simple. So you know now what are the structures going to develop them. So it's easy for me to learn or teach you to here what is the primitive streak development, how the embryo gets formed, and you can also look on this. In this picture, everything is clear. Here is the precaudal plate. That is the future mouth development. Already told you after the formation of buccal pharyngeal membrane is going to rupture. And this is the notochord. Notochord, I already told you. We are going to also see here the development of neural tube. Wait for it. So this is the notochord, and below it there is a primitive peat. We can see here primitive peat, primitive node, and this is the primitive streak, and below it cloacal membrane. They are the future anus, future mouth. The future mouth is developing from the precaudal plate or the primitive streak, of course precaudal plate. And what is the structure going to develop into the future anus? That is cloacal membrane. Simple. The last part is it. And we can see here the primitive peat, primitive node or Hansons node, and the primitive groove. These all are the structure, is the part of the primitive streak. Simple. So we are going to rough it and move further in our discussion. Now you can see there is the Hansons node, is the developed thickening part of the what? That is primitive streak. The proliferation of Hansons node, after that the Hansons node is formed, and this Hansons node is going to proliferate. What the Hansons node is formed, this Hansons node is going to proliferate, and after the proliferation of these Hansons node, cells are forming different cells. That cells are called as pre-notochordal cells. In later stages, these cells are going to form notochord. That's why they are called pre-notochordal cells, and these pre-notochordal cells are going to invaginate, and after the invagination of these pre-notochordal cells, they are going to form primitive peat or the blastopore. Primitive peat formation is how the first Hansons node is formed. After that they gets proliferate, then the Hansons node is forming the pre-notochordal cells, and then they gets forming the primitive peat or the blastopore. How we are going to say? Wait, I am form a diagram for you. So look on this. Hansons node is the proliferation, is going to proliferate, and the Hansons node is forming the pre-notochordal cells, and after that the invaginates and form the blastopore or the primitive cells. Till now we can see, and after that we can also say this Hansons node is going to forming the primitive pit. How they are going to form, we see after few seconds. These are forming the primitive pit. After that they are forming the notochordal process, and then notochordal canal, then they're forming the notochordal plate, and at the last they're forming the definitive notochord or the proper notochord. So the notochord is forming basically the step. They are following the different types of steps, and after these steps at the end we get the notochord. So how they are going to form from the primitive stick? Yeah, from the primitive stick we are going to see here. This is the primitive stick here, and from the primitive stick the Hansons node begins. That is a thickening of the primitive stick, and this primitive stick is going to form a node, that is Hansons node, or we can also say the primitive node. And this primitive node or Hansons node is going to proliferate, and after the proliferation they are converting into the pre-notochordal cells. And these pre-notochordal cells are called as the blastopore, or either they are also called as primitive pit. So these are the same term. What are the same terms? Either they are the blastopore or either primitive pit. So now we are going to see in the picture. We can see here in this picture. Suppose that here you can imagine here. This is our ectoderm. This is our endoderm. And in between them here, all region, there is a mesoderm. In between them, what is it? Mesoderm. Now tell me, between the ectoderm, mesoderm and endoderm, between them there is a neural tube or the notochord is formed. Wait a minute. That is neural tube and the notochord is formed. So how this notochord is formed? This notochord and neural tube is basically present in between them, ectoderm and endoderm, and this mesoderm is not going to line them. I already told you, the cloacal membrane and the prochordal plate, these are the two regions where they are not going to line. So neural plate and the neural tube, we are going to see a notochord, how they are going to develop. This notochord is developed from the epiblast region, which cells primitive streak. This primitive stick are going to thicken Hansons node and then gets proliferated. The pre-notochordal cells are formed, and after that these pre-notochordal cells are developing this, and there's a different steps are happening in between them, and after that finally they gets formed. Now what are the different steps is going to hidden here, and later on they forming the notochord. We are going to see here in this picture. Now you tell me, in this picture what is the upper one? This one is called as what layer? This one is called as ectoderm. This one is endoderm. In between them this region is mesoderm. So now you tell me, ectoderm, mesoderm and endoderm, and something is formed in the epiblast cells. The ectoderm, they're going to modify it here, we can see, and they're forming the blastopore. Here is the one pore is creating, and that is called as blastopore. Simple. Through this blastopore, or the we also can call like this, the primitive pit, they are going to invade inside the mesoderm, and they are moving down, and they are coming to attach with. They are coming to attach in this layer, that is called as endoderm. They are going to attach with the endoderm, the basal part of the endoderm, and they are going to attach through it, and the attachment after the endoderm is called as, this structure is called as notochordal process. First, what is happening here? The primitive stick is going to modify into the Hansons node or the primitive node. They are going to sending some signals inside the endoderm, and these endoderms are, after the reaching these signals, is called as the notochordal process. And this formation of notochordal process is basically after the beginning of blastopore is they're going to invade inside it. They're going to invaginate in the endoderm, and we can see it. How they are going to invade, we can already see. There is a Hansons node. Later on, these Hansons node are forming the primitive pit or the blastopore, and these blastopores are forming the notochordal process, and these notochordal processes are forming the notochordal canal. What is the notochordal canal? What is this? This is the notochordal basically process. This structure is notochordal process here, and this is canalized. Canalized means there is a canal is formed in between them. Suppose that this is the structure is, this structure is called as what? notochordal process. They are going for canalization. Inside it there is a canal is formed. Understand? Inside it the canal is formed. The canalization of the notochordal process leads to the formation of, and now this called as notochordal canal. Notochordal canal. Basically these all are not happening at the same place. They are happening in a step-by-step manner. First it's happened, the blastophore is formed. First blastopore formed. These blastopore are moving down. They are going to fused in the endoderm walls, and they are canalized. After the canalization they are called as notochordal canal, and then further they are going to fuse this notochordal canal. Through this notochordal canal, what is built up here, and you know above the epiblast a cavity is here and a cavity is also here. That one is called as amniotic cavity. I already told you amniotic cavity. Here is yolk sac, and the amniotic fluids, what is present in the amniotic cavity and what is present in yolk sac, the yolk sac fluid, they are going to move across this canal. So they are communicating. There is a transfer of fluids will be occur through it. That is canalization. Now next point we can see here what is going on. After the canalization there is a notochord formation. How the notochord is gets developed, we see. Wait a minute. Now you know where is ectoderm, where is mesoderm, and where is endoderm. So I'm not going to tell you where is ectoderm, mesoderm, and endoderm. These all are hidden here. So now you can imagine, and this is the process here. This and this structure is going to, this one is the another picture. This one I already told you, simple epiblast, that is they are going to form the ectoderm, mesoderm, and endoderm. All the parts are deriving from them. So the upper one is deriving, that is ectoderm. In between the ectoderm and endoderm there is a mesoderm. Simple. That this is the notochordal process is forming like this in the ectoderm. So these both picture are same. This is more differentiated. They are the more view, zoom view, and this is the complex picture of it. So we are going to see further what is happened. There is a canalization is happened in this. I already told you there is a canalization is happened in the blastopore or the notochordal process, and after that the fusion of this with the floor of the notochordal canal. This notochordal canal is going to they are going to fuse with the endoderm. How they are coming? They are forming canal from the ectoderm, and they are coming down, and they are fusing with endoderm. So the surface of endoderm is going to fuse with it. They are coming like this, they are coming like this, and they are going to fuse here with the endoderm. Now you know ecto, and this one is endo here. Through this they are going to fuse here, fusing with this membrane. So it's so simple. After the canalization, the canal is formed, they are going to fused, and next one, the breaking down of the fused parts of the floor of notochordal process and the endoderm. Now once again we can see here, suppose that this is the structure ectoderm, endoderm, and this one is mesoderm, this whole structure. Now I'm going to tell you, from here the blastopore is formed, and from the blastopore the notochordal processes are moving down, and they are going to fuse here in the endoderm part. So the fusion of this endodermal part in this region, the fusion is going to be tell some special name, and that one is called as breakdown of fused parts. What the part of notochordal process, they are coming from the blastopore, they are going to fused with the endoderm layer, and they are going to, after the fusion there is a communication in between. Here is the amniotic cavity and here is the yolk sac cavity. So inside them the fluid is filled, so they are communicating with these canals, and after that there is a fusion, they are forming the notochordal plate. After the fusion with this, in this region they are going to fused. Now you can imagine, if it's coming here and it's going to fuse, it's forming a plate like a structure, and this one is called as notochordal plate. Okay, and this notochordal plate is going to develop here, and after that they are forming a here notochord, definitive notochord, around like a structure in between the ectoderm and endoderm. So it's so simple. Now next we can see here they are communicating with yolk sac. Yeah, simple. The formation of notochordal plate. Yeah, you know now. And this notochordal plate is becomes curved, invaginate, and forming the definitive notochord. How? Once again, this one is, and here they're coming and fused here. They're fusing forms the notochordal plate, and this notochordal plate is going to invaginate like this. Invaginate, and in next step they are going to form this. This is the definitive notochord, and this is the notochordal plate, and this one is notochord. Now the notochord is formed here. This is the ectoderm, and here is the endoderm. So it's so simple. In next section we are going to see about the notochord structures, what they are going to develop here, and the different parts of it. So now you see this Hansons node is going to convert in the primitive pit or the blastopore. Already told you, these blastopore are invading into the endometrium. They are moving down and they're forming the something, that one is called as a notochordal process. And this notochordal process, after the canalization of it, the canal form, they are communicating between the amniotic fluid and the yolk sac. There is a formation of notochordal plate, and this plate is going to invaginate, and after the invagination there is a formation of definitive notochord. So it's so simple, and you can see here definitive notochord is it. Next point, here is the summary, something we see later it, but for now you can come here. The fate of notochord. After the formation of this notochord, this much notochord is formed here. Here the notochord is come now. Suppose that this is the notochord. What is it? What is the role of the notochord? So we can see this is the ectoderm, this is the endoderm, and intradermal mesoderm. You already see the third intraembryonic mesoderm. Intraembryonic mesoderm, we can see these all three layers, the first, the second, and the third. There the three germ layers are forming, and inside it there is a notochord. So what happened to this notochord? This notochord is having some fate. There is a, what is the future of this notochord is basically these notochords, we can see here. These notochords are going to disappear. Then why we form this? And the remnants of notochords we can see in our human body in two places. There is a, in our vertebrae parts, you know the vertebrae, there is a cervical, thoracic, lumbar. This vertebrae is having body, you can see here, and in the body is having a gray part. This region part of the body is called as nucleus pulposus. It is basically remnant part of the notochord. What is the development is occur in embryonic stage of the third week. There is a nucleus pulposus is developed further in the body of the vertebra, and they are the remnant part of basically notochord. And next we can see what is the another remnant part. Why we are going to discuss this. The notochord is forming basically the segmentation of our body. We can see here this nucleus pulposus and apical ligament of dens. You know the dens one is the first vertebrae or second vertebrae. The dens process is present on the atlas or axis. On atlas, and the axis is the second cervical vertebrae on which the dens process is present. And through dens we can see here we can look on it. Here is the dens process, and the dens is present on the second vertebra and of the cervical, and this is leads to the apical ligament. What are the ligament is present here? We can see here these are the apical ligaments of the dens. They present through above, they are moving upwards. These are the apical ligaments. We can also see here these are the apical ligaments are also developed from the notochord. So what are the two structures we can see now in yourself? There is a two structures is the remnant part of the notochord which is present in our body. That is the apical ligament of the dens, and the next point is, you know, that is the nucleus pulposus of the vertebrae of body. Now we are going to see what is the role of notochord. What is the main role of notochord in our body? we are going to see the role of notochord is basically this notochord is going to stimulate. Suppose that this is the our ectoderm, endoderm, and here mesoderm. Mesoderm is basically present like this. I am going to give you some important clear view in your mind. So, and now what we formed just now, here from here there is a notochord, NC we formed just now in previous section. Now you want to form neural tube. So it's also formed in third week. I already told you neuralization and gastrulation process is occurring in this. So this notochord is going to stimulate the ectoderm. You can see properly. This notochord is going to stimulating this ectoderm layer and form the neural plate. They are going to form neural plate here. Neural plate. And after that, from this neural plate, there is a development of neural tube. So now you tell me, who is going to stimulate who is going to stimulate the ectoderm to produce or to stimulate the formation of neural tube? That is basically notochord. So it's so simple. They are going to ectoderm, forms the neural plate, and after they form neural tube. Now we are going to move towards the mesoderm layer. How the mesoderm layer is going to form, and its different types of the mesoderm. We already discussed about the ectoderm, endoderm. Now we are going to mesoderm types. You can see a look on this picture, and you can imagine it by yourself. Here is the neural plate. NP is neural plate, and just now you see neural plate is formed by which? That is by the stimulation of notochord. To which we are going to stimulation? That is the ectoderm. It means the upper one is ectoderm. This whole layer is ectoderm. Just below, which layer is going to synthesize the notochord? Already we see they are originating from the endoderm. It means this whole layer is called as endoderm. Now next section. It's very interesting for you. Now I already told you ectoderm, endoderm, and in between them what is it here? That is mesoderm. And mesoderm is basically two types, intra-embryonic or extra-embryonic. Then intra-embryonic, mesoderm, extra-embryonic we already discussed in the second week. What is going on there? That is extra-embryonic mesoderm, is a splanchnopleudic layer, somatopelvic layer. So now we are going to see intra-embryonic mesoderm, which are situated in between the ectoderm and mesoderm. Sorry, ectoderm and endoderm. So this is the ectoderm here, and this is the endoderm here. And what the structure you are looking here, the red one is all are intra-embryonic mesoderm, IEM. And we can see the first, I'm going to draw by yellow, you can see by yourself. This is the first. There are the three types of intra-embryonic mesoderm. The first, this is the second, and here is the third one. So this is the third, three types of the extra-embryonic mesoderm. The first, the second, and the third. These are the three types of extra-embryonic mesoderm is here. So the intra-embryonic mesoderm, they are the three types that present in between the bilaminar disc or the upper and lower, that is ectoderm and endoderm. And we can see this first one is named as somites. The first extra-embryonic mesoderm is called as somites. These are called as somites. And second one is called as the intermediate mesoderm, and this one is called as splanchnopleudic mesoderm. So what are the three types of mesoderm? The first one is called as somites, second one is called as intermediate mesoderm, and the third one is called as splanchnic mesoderm or the somatic mesoderm. With different types of it, we see it later, but for now I am giving overview about it. So don't worry, if you don't understand it, wait for 10 minutes or 5 minutes, you will understand everything. So let's see. Here is the embryo. I'm going to give you this picture, a very real view. You can see here. Okay. And this is the embryo. This is the human embryo basically. This is the embryo. What happened to this embryo? We can see here. Within the embryo, I already told you, this is the cranial end. Suppose that cranial end. It means this is caudal end. and caudal end formed by the membrane. That one is called as cloacal membrane. Next we can say, and at the caudal end, you know, there is a primitive streak is present. They are derived from the what cells? Epiblast cells. And here is the Hansons node or the primitive streak. This one is the primitive streak. So now you know the Hansons node is going to develop into the notochord, so we are going to remove it, or we are going to see this embryo hole. So what is here? That is the in the cranial end, which part is going to develop? That is the precaudal plate. So this is the precaudal plate, and from the precaudal plate, you know what is developed? That is the oral cavity. So precaudal plate, oral cavity, they are going to give something else. And the precaudal plate, just above it, a structure is going to develop here. That one is called as septum transversum. Septum transversum. This septum transversum is basically formed just from the precaudal plate and above it, and we can see after that we are moving towards the notochord. What we discussed till now, this notochord is forming in the midline between the primitive streak. This is the notochord here, and this notochord is called as notochord, present in the mesoderm. So they are called as the midline of mesoderm, in the midline of mesoderm. And this notochord, just aside of it, there is a mesodermal layers are present. How they are located, we can see here. This is the, I'm going to tell you, wait a minute. Suppose that this is the first part of mesoderm, this is the second part of mesoderm, and this is the third part of mesoderm. This is the third part of mesoderm. So there are three types of mesoderm basically. I already told you now. There is a first one is called as paraxial mesoderm. Second one is called as intermediate mesoderm, and the third one is called as lateral mesoderm. What is the structure you see in this diagram? You can also see here. This is the cloacal plate, cloacal membrane, and from this there is anus is developed later on, and here is the precaudal plate, that is the round one, and after that here is the primitive streak. We already see previously primitive streak, and after that we are going to move towards the notochord. This is the notochord here. You can see this is the notochord here, and just lateral to the notochord there is a mesoderm is present, and that mesoderm is called as paraxial. Paraxial means both the sides, that is paraaxial, that is the from the axis, both the axis side, that is paraaxial mesoderm. Just aside of it from the first one is paraxial mesoderm. Next this structure is called as intermediate. These are called as intermediate. The second one is called as intermediate mesoderm, and just aside of it, the away from the second, that one is called as the third point is called as, we can see, lateral mesoderm. So there are the three types of mesoderm: paraxial, intermediate, and the lateral mesoderm. And where they are located? They are located just aside of the notochord, just lateral to the notochord, aside of lateral cord. The first mesoderm is paraxial. Next one is just lateral to it, there is the intermediate, and just lateral to the intermediate mesoderm there is a lateral mesoderm. So these are the three types of mesoderm. So now hope it's clear, the whole structure is clear for you now. And septum transversum is developing from the prochordal plate. So it's also very important structure. They are going to develop the many structures in our body, the septum transversum. So it's important here. What structures are developing from the septum pellucidum, we see in 1 minute. Wait a minute. This is the embryo here. This is the embryo. We discuss everything in embryo. Now the septum transversum is going to give some components or some structures in our body. What are the structures that are forming from the septum transversum? We are going to look on it. The septum transversum is the structure which is undivided part of the lateral mesoderm. This is the part we see here. This is the septum transversum, and you can imagine after some time when I am teaching you the third part, that this is the structure here. This structure is basically, the third one is lateral mesoderm, and this is the undivided part, means they are not going to separate here. So this is the undivided part of the septum transversum, is undivided part of lateral mesoderm, and the third is lateral mesoderm. Lateral mesoderm is present like this. So you can see like this, the lateral mesoderm here. So they are the undivided part of lateral mesoderm. And next we can see they are the also cranial to the prochordal plate. Yeah, it's true, they are present in the cranial end. Prochordal plate is also in the cranial towards the cranial end. Next point we can see here, they are contribute to the formation of what in the embryonic condition in the third week. There is some structures are going to develop. What are the structures is going to develop from this septum transversum? The first structure is going to develop from there is the diaphragm. Our diaphragm is also developed from here, that is the septum transversum. Next one, the liver. The liver is also developed from the septum transversum. And the fibrous pericardium. The pericardium is having fibrous and different types of layers on the pericardium, you know, the visceral layer and parietal layer and the fibrous. So these all layers, the fibrous pericardium basically here, they are developed from the septum transversum. In the liver, what part is developed from the, what is the main components of the liver? Inside the liver there is many cells, that is the sinusoidal structures, Kupffer cells, that is macrophage of the liver, and the connective tissue, what the tissue is present inside the liver. These all the structures of the liver is formed by the septum transversum. So this is also important, diaphragm, liver, fibrous pericardium. In next point you can see here what we discussed previously. We are going to see here the second one, that this one is the first, this one is the second, and here is the third eye, third O. Third eye is basically inner to the something, and third O is basically outer to the something. O is here outer, I is here inner. So what is this yellow cavity is looking here? This yellow, what is it? So this yellow is basically cavity here. Cavity is formed in the lateral mesoderm. Now you know the lateral mesoderm is the most marginal part. So suppose that this is our embryo and this is precaudal plate, cloacal membrane, this is primitive stick, everything is located here, notochord. From the notochord, first one is, that is paraxial, next one is, that is intermediate, and this whole structure, what is located here, that is the lateral mesoderm. So this lateral mesoderm, inside this lateral mesoderm, a cavity is present. So this, in this lateral mesoderm, there is a cavity is present like this. A cavity is located. In the lateral mesoderm, this cavity is divided the lateral mesoderm into two parts. You can imagine, when something is entered in a cavity, in a whole structure, then its rod is going to divide into two parts. Something is going to, this, suppose that this is the hand, when you are moving pen inside it, it's going to divide in the one half and second half. So it's divided into two parts basically. So same here, when there is a, suppose that this is the mesodermal structure and we are putting here something, then it will be divided into two parts, 1, 2. So the two parts is divided. So same here, they are going to divide it into two parts. We can see here one structure is inside it and one structure is outside it. So this structure is, suppose that 3, it means 3I inner and 3 outer, and 3 inner, 3 is the name is in lateral mesoderm. So you can say like this, there is a lateral mesoderm outside the cavity, or second one is lateral mesoderm inside the cavity. The outer part of the lateral mesoderm and the lower inner part of the lateral mesoderm. So this cavity is going to divide. Now you can see here the lateral mesoderm is divided into 3I and 3O, that is the inner part and outer part due to a cavity. And the first mesoderm is, what is it? Now I'm going to give you some brief about it. The first one is paraxial mesoderm. Okay, first mesoderm is forming the paraxial mesoderm, and this paraxial mesoderm is just a side of the notochord. So they are going to develop some structures in our body, and they basically form somites. What is somite? What are the forming somites? What they are structure going to develop from the somites? We discuss everything later after 10 minutes. So for now, we have to memorize this paraxial mesoderm. What is present just aside of the what? Just aside of the notochord. This is called as the, they are going to form in future somites in the future of the third week. So next one, intermediate mesoderm. Just remember, just aside of it, there is a intermediate mesoderm is present, and they are going to develop a urogenital system. The whole urogenital system is developed from the structure, that one is called as intermediate mesoderm. The intermediate mesoderm is the structure which is going to develop urogenital system, and urogenital system is composition of two system, that is urinary system and genital system. It means reproductive system. The urinary system, what the structures are included? The kidney, ureter, bladder, these all are the structures are present. So next one, what is the structures in our reproductive system? Testes. In female there is ovaries, mesonephric duct, they are the kidney development or the paramesonephric duct. So these all structures are developing from the urogenital system basically, and they are developing from which mesoderm? That questions are coming. That is the intermediate mesoderm. It is the part of which mesoderm? That is intraembryonic mesoderm or extraembryonic mesoderm. You know now, they are the part of lateral mesoderm. So these are the structures from where they are going to develop. The urogenital system is developed from the intermediate. Now we are moving to the lateral mesoderm. Now you know the lateral mesoderm is having two parts, that is inner and outer. Same we discuss in the extra embryonic mesoderm. There is a outer and inner part. The outer one is called as somatic and parietal extra embryonic mesoderm, and the inner one is called as somatopleuric extra embryonic mesoderm. So same as here, the inner one is called as a splanchno-pleuric layer of lateral mesoderm, same, and the outer one is called as somatic or parietal layer of lateral mesoderm. So it's so simple. And this lateral mesoderm, what we are going to discuss here, they are forming a cavity inside it, and the outer and inner. The outer one is the somatic or parietal mesoderm or the lateral mesoderm, and the inner one is a splanchno-pleuric layer of the lateral mesoderm. So is this cavity is lining by these two membranes or these two mesoderm. Now these cavities are what? So in our body different cavities, do you know about it? The abdominal cavity, thoracic cavity. So these all cavities in our body, what are it? So the cavities like pleural cavities, pericardial cavities, peritoneal cavities, these all cavities are lining by the visceral and parietal layer, you know from there. The in the our abdominal cavity you will discuss like this, the peritoneum or the parietal layers. There is a parietal and visceral layers of the something. So this parietal and visceral layer is coming from here, from the lateral mesoderm. They are derivative of the lateral mesoderm, and the splanchno-pleuric part or the somatopleuric part or the somatic part or the parietal part. We can see now you can also know there is a in heart pericardium, and in the lungs there is a pleural layer, and the peritoneal cavity. These all are formed by the lateral mesoderm. So the body cavities are formed by them, and the splanchno-pleuric layer is going to splanchno-pleuric layer, that is the innermost layer of the lateral mesoderm, is going to develop something in our body, and that is cardiac muscles, smooth muscles, and the visceral layer of body cavities. What the cavities we discuss? Pleural, pericardial, and the peritoneal. These all cavities is having visceral layer. The innermost layer is formed by the splanchno-pleuric layer of mesoderm, and they are also forming the cardiac muscle and a smooth muscle. So that questions are coming. The smooth muscle is formed by the which mesoderm? So they are the splanchno-pleuric layer of lateral mesoderm. Next question, the outer part, that is somatic layer of the lateral mesoderm, they are forming the appendicular skeletons. Appendicular means basically appendicular means basically appendages, and appendages means basically our limbs, that is upper limb, lower limb. These are so the bones of all the upper limb and lower limb. The bones of the upper limb and lower limb or the appendages are forming by this layer, which is it? That is somatic, means outer appearance. Somatic mesoderm, somatic lateral mesoderm, somatic part of the lateral mesoderm is forming this, and there is also parietal layer is formed by the parietal layer of body cavities is formed by the somatic part, or the somatic layer of lateral mesoderm. So now you tell me, what are the two division of the lateral mesoderm? Somatopleuric and parietal. Parietal one is forming the parietal boundary or the parietal body cavities part, or the parietal boundary of the body cavities. Next one, they are also going to form appendicular appendages or the appendicular skeleton. It means they are going to form bone. All the bones of our tarsals, metatarsals in the foot, or the carpals, metacarpals, or the phalanges, maybe distal, proximal, or all the phalanx. They are the carpals, tarsals, or the humerus, ulna, radius. These all bones are formed by which mesoderm? That is lateral mesoderm. Which part of lateral mesoderm? There is somatic part of lateral mesoderm. This is the most important here. Next, we are going to see somites. Now we are studied detail about one by one. Now you tell me, somites are forming from the which part? We can see here. Here is the somite. It is formed from the proximal mesoderm. So what is the structure going to develop from the somite? So you can see here the somite. The somites structure is developing. What are the structures are developing from the somites? First, somite is the paroxysmal mesoderm. It is true. It means they are located somewhere here. That is mesoderm. That is intraembryonic mesoderm. I already told you, intraembryonic mesoderm. Now you see here, this is the ectoderm, this is the endoderm, and this is the neural tube. Already told you, it is also neurulation is also happening in the third week, and the notochord is forming from the there, you know now. See, those notochords and neural tubes are present in between, and these all are present in the mesodermal part, and this layer here, what is forming here, this is called as somites. The somites are the paroxysmal mesoderm, and they are divided into two parts. This structure is divided into two parts, and the innermost part is called as this structure. This whole structure is called as somites, and this whole somites is going to divide it into two parts. Now you tell me, what are the two parts of somites? The inner one is called as a sclerotome, and the outer one is called as dermamyotome. Now you tell me, what are the two types of somites? There is a sclerotome and dermamyotome. Simple. The sclerotome is going to bound. This sclerotome, this is the region of sclerotome. This is the sclerotome here, you know now. This is the sclerotome basically. So the boundary of a sclerotome is going to cover, or they are going to bonding. This is the boundary of a sclerotome is going to cover this structure. What is the structure inside the sclerotome now? The boundary of a sclerotome, neural tube and notochord. So these two structures are present inside the sclerotome boundary. So we discuss later everything. For now, it's okay. Now you know the dermamyotome and sclerotome. We can see in this picture also. Look on this. So this is the ectoderm here. This is the ectoderm, this is the endoderm, and this is the somites here. Somite divided into inner part and outer part. The inner one is called as sclerotome, and the inner one is called dermamyotome. And these are the somites are developing from the paroxysmal mesoderm. So now you know. Hope you understand everything. If any query, you can ask me on social media side. So now you can see on the surface ectoderm, you can also understand this picture. So surface ectoderm is developing, we see later. This is the ectoderm, is developing the surface ectoderm, we see later how they are going to develop. Now we are only discuss about the mesoderm or the somites. So these are the somites, these whole structures, they are going to differentiate it into two parts. The somites are going to differentiate into two parts, the outer and inner, that is the sclerotome and dermamyotome. And these structures are the notochord is here, and this one is neural tube, we can see, and inside the neural canal. So these are the neural crest cells, that is 4 type of germ layers is present here, just above the neural tube. So we see later, but now you know this diagram. So these somites are basically, they are the segmentation, they are forming like this bead-like appearance. These somites are forming in the pair basically, they are forming in this like manner. So somites are forming in the beaded-like appearance. So this beaded-like appearance of somites are first time forming in the cranial region, our head region, and this is called as first pair of somites. First pair of somites, when it form at the third week, and the somites are developing from the paroxysmal mesoderm, and this paroxysmal mesoderm are forming the first pair of somites, and the first pair of somites are going to develop in the occipital region of our brain. And occipital is behind side, these are the occipital bone, and they are going to develop. Now you know, okay, there is a dermamyotome and a sclerotome, there are two types of somites. So the some structures of the somites are going to develop the occipital bone or the occipital region. So these are the happens on the 20th day. The first somites are developed on the 20th day of the embryonic development, on the first 20th date, and we can say next 3 pairs are added each day. It means the one pair is forming on the 20th day. Now you can imagine one pair of the somites are formed on the 20th day, and the after 20th day there is a 21st, 22nd, 23rd, like this. This all day there is a 3 pairs are going to add. It means here is the 1 pair and 3 pair is going to add after that. It means on the 21st there is a 1 plus 3, it's 4 pair. On 22 again 3 pairs is going to add, that is it is equal to 4 plus 3, that is 7 pair. Once again on the 23rd day there is a 7 plus 3, that is 10 pairs. So their number are somites going to increase, and it is increased up to 5th week. The number of somites will be increased up to 5th week. On the 35th day, or you know now, on the 35th day, or the we can say the 5th week, there is a 45 pairs of the somites are formed. And what is the role of these somites? We discuss it within 2 minutes, but for now you can see here these somites are formed 45 in number approximately. There may be 42 to 45 like this, but they are from 45. And what is how they are going to attach? You know, 3 pairs are adding each day, and on 35 day they are forming up to 45 stage. What are the formation from the somites? What are the structures? And you know the sequence is like craniosacral sequence, the formation of germ layers, same as here. The formation of somites are the same. The formation of somites are the which, how many pairs? 45 pairs. They are going to develop like this. There is the first pair forms the occipital structures, second one is cervical, then thoracic, then lumbar, sacral, and the coccygeal. So these are the vertebral column lesions. They are going to develop from the somites. So these are the structures which are following from the development from the first somites. Now we are going to see the some somites in the occipital region and the coccygeal region are going to disappear. What is happened? Some somites which are developing in the occipital region and the coccygeal region will be disappear. Only in which region? In the occipital region and in the coccygeal region. All other are same. So how many somites are there? 45 pairs. So out of them, the sum of the somites are going to disappear from the occipital and the coccygeal layer, and we can see here, and at the end of the 5th week after the development, they just 37. They are just approximately 37 somites are present in the embryo. So they are forming 45 after, or 42. They are going to disappear or degenerate, and there are only 37, approximately 36 or 37 somites are formed at the after the 5th week. After the 5th week, the whole embryo, we can see only 37 pairs of somites, and they are going to develop the different structures from them. We can see here the number of somites vary up to the 5th week. It means till the 5th week they are going to develop. So there is an increase or decrease in the number of somites will be depend on the week. So after the 5th week it will be the constant, but before the 5th week it will be maybe varies or the gets different number. So the remains constant after the 5th week. It is the same. Now you can see there is some structures we are going to discuss here. Somites differentiation, and here is the sequence, one sequence, and this. First they are going to develop A, then B, then C. So A structures you can see, and you know about it. This is the notochord here. This is the neural tube here. Let me take some other color. Here neural tube, here notochord, and this is the somite here. This whole structure is somite. So you can see somite. Somites are going to be divided into two parts, sclerotome and dermamyotome. Sclerotome is inside and outside is dermamyotome. Same here. Now there is the sclerotome is same, but the dermamyotome is divided into dermatome and myotome. The dermamyotome is outer part and the myotome is inner part. So these are also divided, dermamyotome into dermatome and myotome. So what are the structures developing from the sclerotome and what are the structures developing from the myotome or the dermamyotome? We see now. So we are going to see about the division of, see this structure, you may understand. This is the somite, you know now. And the somite is divided into two parts. Okay, the inner one is called as sclerotome. and the outer one is called as dermamyotome. This dermamyotome is going to divide it further into two parts, the dermatome and inner one is myotome. Understand? So the dermatome, myotome. Now we are going to see about these structures. So the parts of somites, you see there is a structures. We can look on this. This is such an important here. Now this is the ectoderm. We can see a ectoderm, you know. This is the endoderm, and this is the whole structure is somite. They are divided into two parts. Here is the sclerotome. Here is the sclerotome, and here is the dermamyotome, and here is the sclerotome. And this sclerotome is surrounding the neural tube and notochord. This is the structure. And now you know the ectoderm is formed from the ventral part. This is the ventral here. What the structure forming? The front of the notochord, they are called as ventral part, and the posterior to it they are called as dorsal part. So this is the dorsal part of the sclerotome, and this is the ventral part of the sclerotome. This is the dorsal part of the sclerotome, and this is the lateral part of the sclerotome, because the sclerotome is surrounding and covering the neural tube and notochord. So now you know three parts of the sclerotome. There is the ventral, here is the lateral, and here is the dorsal. So three parts. This ring is having three parts. The dorsal according to notochord, the ventral, and here is the lateral. So these are the three sclerotome part, because this round structure is a sclerotome. So dorsal sclerotome, lateral sclerotome, and ventral sclerotome. So hope it's clear for you now. These are three sclerotome. Now moving another direction, we are going to see now what are the sclerotome parts. Now you see there is a ventral, dorsal, or the lateral sclerotome. What are the structures going to develop from them? So we can see the somites first. The somites is divided into a sclerotome, dermamyotome. This sclerotome forming the axial skeleton. Here most important point. This sclerotome, we can imagine now. They are divided into two parts, and here is the sclerotome. This sclerotome forming the axial skeleton. Axial means our body here from the shoulder to the we can say like the waist region. These are the axial region. And what is the bones are present here? That is the vertebrae basically. So these all the vertebra are developing from the sclerotome. Simple. And somites. And somites are developed from the paroxysmal mesoderm. It is true. Hope it will be true. Next we can say the sclerotome. Sclerotome, the first point, ventral, lateral, and dorsal. Already told you. The ventral part of the notochord, front of it, the sclerotome part is called as ventral sclerotome, and the ventral sclerotome going to develop some structure. That one is called as body of the vertebra. So now you tell me, sclerotome is forming the vertebra. It's simple. It's already written here. Sclerotome is going to form vertebra. But what is the different parts of vertebra? You know, there is a body, there is a spinous process, transverse process, vertebral canal, or the pedicel. Different parts are present in the vertebra. So there is a different parts for the different parts is going to develop from the different regions of a sclerotome. So we can see the sclerotome is having ventral part is going to form the body of the vertebra, this region of the vertebra. We can see here. This is the body of the vertebra is formed by them. Body. Next, the lateral will form the arches. The lateral will form the vertebral arches, and arches, you know, there we can see the vertebral arches here. These arches are formed by the lateral, and now we are moving to the dorsal sclerotome forming the spine. All the spines we are going to look on the vertebrae. Here is the spinous process, transverse process. These all are the spines of the vertebra. So these all are forming from the dorsal sclerotome. So now you know the vertebra are going to develop from the sclerotome, dorsal, ventral, and the lateral. And what part of the vertebra is formed by the what different parts of the sclerotome. It's very easy and simple. So don't think. Next you can see, suppose that if there is any deformity, in the formation, what type of deformities. There is a failure of fusion. You can see here the failure of fusion of ventral sclerotome. Then what happened? You can see here in this picture. This is the ventral sclerotome. Yeah, this is the ventral sclerotome. If this sclerotome is not going to fused, it means what happened? The ventral sclerotome forming what? They are forming the body of the vertebra. Yeah, it means they are not going to fuse. It means the body will be formed only half. Simple. When the sclerotome is ventral sclerotome is not working and it will be forming the body of the vertebra, so it will not going to form the body of vertebra. It means the body of vertebra will be hemi. It will be half because it form half and they are not going to fuse. So we can see here the body of vertebra is forming the half. We can see here this term is hemi means half, vertebra means half, vertebra adha. We can see here the hemi vertebrae. This is the hemi vertebrae here. Wait a second. This is the hemi vertebrae here. Hemi vertebrae. This type of vertebrae is formed when the ventral sclerotome is unable to fuse. Next deformities we can see spina bifida anomaly. In this, the failure to fusion of dorsal sclerotome. What is dorsal sclerotome? Where it is fusion? In this region. If in this region the fusion is restricted, the fusion is not happening, then what the dorsal sclerotome is going to develop? That is spine of the vertebra. Then a spine of the vertebra is not formed. Simple. So this is called as spina bifida. When the spine is not formed, they are not going to fused. Basically, so when the spine is not going to fused, after the fusion they are forming the one spine, suppose that, but they are not going to fuse. It means they are forming like this or this. So it forming the two structures, two spine. The spinous process is not going to fuse of both the sides, so they are forming the two spine, and that's called as spina bifida. There are two structures of the spine. So we can see here. Now next structure is dermamyotome. Dermamyotome, you know, they are divided into two parts, dermatome and myotome. Dermatome. Dermatome. Look on it, the dermatome is it. Here is the dermatome basically, and here is the myotome. Myotome is basically going to develop all the skeletal muscles, the muscles which are present on our bones or they are attached to our bones. These all the muscles are formed by the myotome, and the dermatome is going to develop the derma means dermis of the skin. Now you know the skin is having layers, different layers on the skin. The uppermost layer is called as epidermis. Just below it they are dermis. So the dermis is formed by the dermatome. So the dermatome forming the dermis of the skin, then which layer is going to form the epidermis? The epidermis is formed by the surface ectoderm. We discuss this later. For now you have to memorize the dermatome is forming the dermis layer of the skin and epidermis is formed by the surface ectoderm. So it's so simple. Hope you understand about it, everything. The dermatome is divided into myotome and dermatome. Dermatome from dermis of the skin. Suppose that in any patient what happened? The upper limb bones are formed by the, you know, just before we say the somatic layer is forming the bones of the upper limb by the lateral mesoderm. Now you can see the muscles of the upper limb is forming the biceps muscles, triceps muscles, biceps brachialis muscles or the radialis muscle. These all are the muscles in our hand, upper limb. They are forming by how? They are formed by the myotome of the somates. And these are basically derived from the paraxial mesoderm. So the lateral mesoderm is forming the bone, but the paraxial mesoderm is forming the muscles. Spina bifida anomalies, we are going to see what are the deformities which happen. Suppose that you can look on this picture, you understand everything. This is the normal condition. This is the normal or a spine. And we can see in the spine, this is the vertebral body or the vertebrae. This is the spinal canal region. In this there is a here. These are the nerve, a spinal cord, nerves are here. And there is a opening. If there is a spina bifida, means the spinous process is going to absent here. It means there is an opening here. And due to this opening, they are called as the bifid like spines. That's why they are called as spina bifida. Next structure, what is going on? There is a meningocele. meninges cell means basically swollen. We can see the meninges swollen. What the meninges are present in our spinal cord, the dura mater, equinoid, pia mater. These all meninges of the membranes are going to come out through this, and they are come out and they form a balloon-like structure, that is meningocele. And in the next, myelomeningocele, what is happen? These nerves are coming in the cavities. These nerves are moving outside, so they are myelomeningocele. You can see here the failure of fusion of dorsal sclerotum. I already told you. Okay, suppose that in the dorsal sclerotum here is the structure here. This is the dorsal sclerotum. This is the ventral sclerotum. Ventral sclerotum, dorsal sclerotum, and this one is lateral sclerotum. So if there is no fusion in the dorsal sclerotum, there is a development of spina occulta bifida or the bifida occulta. Same meningocele is also due to the fusion deformities in the sclerotum dorsal. Same meningocele in the dorsal sclerotum. So these all three diseases are from the dorsal sclerotum fusion are interrupted here. So now you know the spina bifida occulta, meningocele or the meningomyelocele. These are the three diseases which are related with the defect in the fusion of the dorsal sclerotum. So there is a dorsal sclerotum is not going to fuse, and these three diseases are appear. So what happened in each disease, we are going to look on it. So there is a tuft of hairs are appear on the back. If any person is suffer from the spina bifida, the person is having on the back of that person there is a hairs are present. The tuft of hairs are present on his body. We can look on it, and you can see here these all hairs are appear in the spina bifida occulta patient. So next we can see there is also the failure of dorsal sclerotum, the fusion of the failure of dorsal sclerotum, and there is a protrusion of meninges over the defect. Where the defect is happen, there is a protrusion of these all meninges. So this disease is called as meningocele. Next one we are going to see meningomyelocele. There is a failure of again dorsal sclerotum, and the protrusion of meninges with spinal cord. These spinal cord are also coming outside. So we can see in this picture, these are the spinal cord. These spinal cord are also moving outside in this meningocele cavities. So this is called as meningomyelocele because there is a spinal cord is present here. Spinal cord. So these are all three diseases are associated with the defect in the dorsal sclerotum basically. So it's now easy for you to memorize all these diseases. Now we are moving towards the development of smooth muscles. There is some exceptions here. There is mainly the development of smooth muscles in the different parts. There is a splanchnopleutic layer. All the smooth muscles are derived from the splanchnopleutic layer of the lateral mesoderm. But somewhere in our body, in the some special region, they are not derived from the lateral mesoderm splanchnic layer. They are derived from the different germ layers. So now we are going to see the first one, the smooth muscles over the dorsal aorta. Our thoracic aorta, abdominal aorta, and the ventral part or the dorsal part of the aorta is going to form by the smooth muscles, and these smooth muscles are not derived from the splanchnic part of the lateral mesoderm. Previously they are derived from the paraxial mesoderm. So it's from the old books they are written. So it's controversy here. For to memorize that the paraxial mesoderm is going to develop the dorsal aorta, smooth muscles of the dorsal aorta. Now it's simple, just same as the splanchnopleutic layer of the lateral mesoderm. Next we can see here the smooth muscles over the iris, dilator pupilli, sphincter pupilli. These are the muscles which are present in our iris muscle. These are the iris muscle are going to develop not from the lateral mesoderm splanchnopleutic part. They are going to develop from the neuroectoderm. So it's exception here. Next one, smooth muscles over the sweet and mammary gland. Sweet and mammary gland smooth muscles are going to develop from the different parts, and that is the not from the lateral mesoderm splanchnopleutic part. They are going to develop from the surface ectoderm. So this is also exception here. Next, smooth muscles over the pharyngeal arches or the pharyngeal arch arteries. What is the artery located in the pharyngeal region? These all are the smooth muscles in that area is going to develop from the neural crest cells, not from the lateral mesoderm splanchnopleutic part. So they are the four exceptions. You have to memorize it and learn by heart from what these layers are going to differentiate and they are going to form. So next part, we also know about that in third week there is a development of gastrulation, yeah, that is the formation of germ layer, we know, and that is the different types of germ layer we know now, and how the structures are developing from the germ layers. The different types of germ layers are going to form the different structures. Now we're going to see the neurulation. Neurulation means basically the formation of neural tube, you know it, okay, the formation of neural tube, that is neurulation, same, and there occurs in the third week. We discussed this in the development of central nervous system. For now we're leaving it, but this is also happen in the third week. The neurulation we discuss in the later videos about it. So you can see next, angiogenesis. Angio means vessels, blood vessels basically, and genesis means formation. So now you know angiogenesis means formation of blood vessels. So we are going to see how the blood vessels are formed. The blood vessels are also formed in the third week of the development phase. So the angiogenesis means the blood vessels formation, and the formation of blood vessels I already told you. There is the angio means blood vessels, genesis means formation, simple. So it is the formation of blood vessels here. It is also occurs in third week. The third week's gastrulation, neurulation, angiogenesis, hematopoiesis, these are occurs in the third week. And the mesodermal layers, they are also differentiated from the mesodermal layers, that is the blood vessels are formed. So the blood vessels are formed from the mesodermal layer, and which part of mesodermal layer now we are going to say. Now you know the extra embryonic mesoderm from the third part. The extra embryonic mesoderm is having also two parts. There is a splanchnopleudic layer and the somatopleuric layer. The splanchnopleudic layer is first time giving the blood vessels. The splanchnopleudic layer, it means the inner one, they are starting the formation of blood vessels, the first, and the differentiation of a splanchnopleudic layer of extra embryonic mesoderm is the first time forming blood vessels. Later on, the blood vessels are also formed by the somatic layer of extra embryonic mesoderm. EM is your extra embryonic mesoderm. So the extra embryonic mesoderm, somatic layer forming the blood vessels later on, but initially it is formed by the, that is splanchnopleudic layer. So and this somatic pleuric layer is going to differentiated and forming the blood vessels, and these blood vessels are seen in tertiary villi basically. Now you know the primary villi is consist of the syncytotrophoblast and cytotrophoblast, that is syncyto and cytotrophoblast is forming the primary villi. The syncytotrophoblast and cytotrophoblast plus extra embryonic mesoderm, they are forming the, that is mesoderm, is these three are forming the secondary villi. In the tertiary villi there is a blood vessels are included. Now we can say the tertiary villi is containing the blood vessels. As I told you, they are forming by the splanchnopleudic layer, and later on they are forming the somatopleuric layer. They are the somatopleuric layer of the extra embryonic mesoderm, cytotrophoblast and syncytotrophoblast. These all four are combined together forming the tertiary villi, and they are formed by the end of the third week on the 21st day. So now we are going to see angiogenesis sequence, how the angiogenesis is going to form. Now you know the first, it is formed by the splanchnopleudic layer. So a splanchnopleudic layer it is going to form, and a splanchnopleudic layer, that is mesodermal layer, splanchnopleudic extra embryonic mesoderm layer. So it is the and later on they are formed by the somatopleuric layer, and basically they all are composed of extra embryonic mesoderm. They are going to differentiate, and these are blood vessels formed in the tertiary villi. We can see it. After these blood vessels is going to form, there are some blood vessels are also forming, but in the intra-embryonic mesoderm. So the first extra embryonic mesoderm, later on formed by the intra-embryonic mesoderm. The first formed by the extra embryonic mesoderm is basically splanchno, then parietal or the somatic layer, and after that the intra-embryonic mesoderm layer. So they are the sequence of the formation of blood vessels. They are first formed by what? They are first formed by the extra embryonic mesoderm. Which part? A splanchnopleudic layer. A splanchnopleudic layer. Next one, they are formed by the extra embryonic mesoderm, somatic layer. Later on it is formed by the intra-embryonic mesoderm. So it is the sequence here, the formation of blood vessels in this way. So next point we can see here, the anastomosis of blood vessels in the extra embryonic mesoderm and intra-embryonic mesoderm. At that level, when the extra embryonic mesoderm blood vessels are going to finish the synthesis of the blood vessels, they are going to is anastomosis, means there is a connecting link between them, that is interembryonic mesoderm. So it is due to the anastomosis here. Now we are going to see what is the applied anatomy. The angio means, once again, there is a blood vessels, and oma means basically tumor. So what does it mean? The tumor of blood vessels, simple, the tumor of blood vessels, and it is the origin is mesodermal. Now you know the angiogenesis is happened in the mesodermal origin, so it is the mesodermal type of tumor. The tumor is also, we discuss everything in the pathology, that is the tumor is basically two types. Either it is mesenchymal origin, or it may be epithelial origin, or the connective tissue type origin. So it is the mesenchymal or mesodermal origin. Now we are going to see the next step in the third week. What is going on? There is a blood cells are synthesized, that is hematopoiesis. And what are the formation of blood cells? They are carried out up to the 5th week. Till the 5th week, they starting from the 3rd week, and up to the 5th week, they are synthesized by the splanchnopleurotic layer of extraembryonic mesoderm. So once again, the splanchnopleurotic layer of extraembryonic mesoderm is also synthesizes the blood up to 5th week. But just after the 5th week, it will synthesized by, from the 5th week or after the 5th week, it is synthesized by the liver, thymus, spleen, bone marrow, LTSP. That is the LTSP, that is the liver, thymus, spleen and the bone marrow. They are also synthesizes the what? Blood cells after the 5th week. But before the 5th week, the development of embryo, it is synthesized by the splanchnopleurotic layer of mesoderm. Next we can say the liver. The liver is the only hematopoietic organ. They are synthesizes the blood cells till 7 months. After that it will stop. So the liver is the hematopoiesis occurs only till the 7th month. What is the placenta development here? Now we are going to see placenta, how the placenta is going to develop. The placenta is basically having two components. We can say like this. The placenta is formed by the two components structure. They are meeting together and they form this structure. So the one is maternal component, the other is fetal component. So suppose that this is the placenta here. So we are going to remove it. So one is maternal component. It means some part of the maternal means mother is participate in the formation of placenta, and some part of the child are the fetus. What is the fetus is developing during that time? That fetus is having some part is going to embedding to each other. They are both invaginates in between them, and they forming the placenta here. So the placenta is having two components, that is a maternal and fetal. That is maternal component and fetal component. What is the name of maternal component which is participate in the formation of the placenta? Maternal component is basically decidua basalis is the term most important here. That is the decidua basalis. Decidua is the term is basically used in pregnancy. Decidua, we can see here. Decidua is basically is seen in the pregnancy, and they are called as uterine endometrium. Suppose that here is the uterus, and uterus is having inside, they are lined with endometrium. So this endometrium, this endometrial wall is called as decidua basically. So the endometrial glands are going to proliferate. You know now, the endometrial glands, endometrial blood vessels are going to proliferation is happen there. So it will be like decidua basalis is having two parts. Decidua is having basically, three parts. Decidua basalis, decidua parietalis, and decidua, we can see here, capsularis. Wait a second. So decidua means basically the uterine endometrium. Simple. It means the uterine endometrium is going to develop in the different regions. How we call it, the capsularis, parietalis, or the basalis, we are going to see later. But for now we can see what is the maternal component, decidua basalis. What is it? We see later. And next one is fetal component is chorionic villi. Simple. You know, chorionic villi is also called as chorionic frondosum. That is chronic frondosum. So chronic frondosum either chorionic villi. These are the two components, or they are the same name. They are the fetal component and the decidua basalis of the maternal component. They both are come together and they forming placenta. Simple. So we can see here, this is the fetal component, that is tertiary villi, and tertiary villi is having venous capillary, that is the blood vessel. Simple. So you can see also in this picture, here the tertiary villi, secondary villi, and primary villi. They are the three villi. and inside it we can see chorion. We are see chorion, you know now, chorionic villi is formed. Here is the lowest most layer is the, you can see, extra embryonic mesoderm. Just above it cytotrophoblast outside, and inside it syncytotrophoblast. So it is the sequence. After that these cells are in the primary villi, you know, there is no any blood vessels, they are simple. In secondary they are going to start vascular lacuna is formed, you know, vascular lacuna inside which the maternal blood components are present, so it will be the secondary. In tertiary there is a blood vessels are formed, that is angiogenesis will happen, chorionic villi, and there is a decidua basalis, it is the maternal component here. Here is the maternal component, so in this maternal component they are going to invade. So it's so simple. Next we can see, suppose that this is the uterus in this picture, and we can see here is this structure is a finger like projection, that is the chorionic villi. And you know the chorionic villi is also called as chorionic profundus, that is frondosum. These chorionic villi are going to invade in the maternal component. This is the all uterus is part of mother, so it is the maternal component. And you know the endometrium is whole lined by the inside there is a myometrium. So this myometrium, endometrium, these all are the wall inside the myometrium. This is the myometrium. In the myometrium they are going to, in the endometrium, two parts. Decidua basalis is the part because it is the base of the connecting base, it is the connecting base of the fetus and mother. So there is a finger like projection in between them, and they are connecting, they are invading to each other. The mother or fetus are going to invading, they are meeting to each other in this area. That's why they are the basalis, they are the base of the fetus development. And just behind it there is a parietalis, that is parietal region, decidua parietalis. And just below that, this one is the cavity of uterus. At the side of the cavity of uterus, that region is called as decidua capsularis. They are looking like a capsule here, that's why decidua capsularis. So decidua basalis towards the base, that is chorionic villi, chorionic frondosum is formed, and just beside it there is a chorionic parietalis, and below it chorionic, to the side of cavity there is a chorionic capsularis. So these are the three types of capsule, or we can say like the decidua basalis, decidua capsularis, decidua parietalis. And now we are going to see what are the main roles of it. So decidua basalis is connecting the fetus and myometrium of the uterus. And next they are the maternal component of placenta. Basically they are the maternal. Decidua basalis is a maternal component. Now decidua capsularis is present in between the fetus and uterine cavity. Already told you, between the cavity and uterus. And the decidua parietalis is the rest of what the decidua is left, what the myometrium is left after the capsularis and basalis. They are called as decidua parietalis. So this is the sequence of forming everything in the third week of the fetus development. And about the neurulation we discuss in the development of central nervous system. So we are going to finish here our third week development, and here also end of the general physiology. So, general embryology is also end. In the next section, in the general section of our embryology, we are going to see in a special topic about the embryology. So there is a different types of organ development, different types of system development, how the system is going to develop from this endoderm, ectoderm, or the mesoderm, or the neural crest cells. There are different parts are developed. And before it, we are going to finish it. I already told you there is a summary section is here. You can look on this. We didn't study till now, but we can see the ectoderm is divided into surface ectoderm, and this surface ectoderm is present outside and inside. What the structure, the surface ectoderm is forming outside structure and inside structure. There is the outside structure is epidermis of skin, nails, hairs, sweat gland, sebaceous gland, mammary gland, parotid gland, and adenohypophysis, pituitary gland. These all are the structures are formed by the surface ectoderm. Next, inside there is a outside, the present outside we can see lens, conjunctiva, lacrimal gland, tarsal gland, epithelial. So these all are the external urethral orifices or the anal. These all are the surface ectoderm origination. So you have to memorize directly. These things we have to memorize directly. What are the examples of the surface ectoderm? So these are the examples of the surface ectoderm. We have to know by heart. Next one, neuroectoderm development. We discuss these things in the neuroanatomy, but for now you have to memorize it. there is a neural tube in which there is a brain and spinal cord development. The brain and spinal cord, astrocytes, there is a supporting cells or the glial cells, they are also developing astrocytes, oligodendrocytes, ependymal cells. These all are developing from the neuroectoderm. Next point, we have to know about the eyeball. There is a retina, optic nerve, epithelial cells, ciliary bodies, iris muscles. So these all are the structure also developed from the neuroectoderm. So ectoderm is divided into two parts, surface ectoderm and neuroectoderm. The neuroectoderm is basically for the nervous development, but we discuss in the neurology neuro development of the central nervous system. Next we can see here in the central nervous system and peripheral nervous system, the myelination is done by the oligodendrocyte swan cells. Neuroectoderm derivative is the central nervous system, that is oligodendrocyte cells. In the peripheral nervous system, the development is done by the swan cells. But what the swan cells production is done by the in condition, that is the neural crest cells. So it is a 4 type of germ cells. Now endoderm development, we can see they are the gut tube, all the foregut, midgut, hindgut. These all the foregut, midgut, hindgut is going to develop from the endoderm part. So the endoderm, you know now from where they are going to develop and what are the parts. There is a foregut, hindgut. So we can see here foregut, there is a entire epithelium of GIT, epithelium of respiratory tract, liver, hepatobiliary tract or the apparatus, pancreas. These all are the foregut. Now hindgut, there is a anal canal, pectineal line, bladder, urethra. These all are the hindgut. So these are the structures which are going to develop from the endodermal in origin. So you have to memorize it directly. Next we can see what is the origin of different parts of the endodermal pouches. The endodermal pouches, there is a tonsillar fossa, thymus, parathyroid gland, or the epithelial lining of middle layer and the stretching tube. These all are the structures which are derived from the endodermal pouches. Next one, neural crest cells. What they are going to develop in our body, we can see they are the collection of cell body in the central nervous system is the nucleus or the nuclei, or the collection of cell body in the outside the sinus is the ganglion. So the nucleus is developing from the neuroectoderm, but the ganglion are developing from the neural crest cells. The cranial nerve ganglia 5, 7, 9, 10, or the dorsal root ganglia, sympathetic root ganglia, or the parasympathetic, these all are developing from the nervous crest cells or the neural crest cells, we can see. And all the ganglion, swan cell, melanocytes, adrenal medulla, parafollicular cells, connective tissue, head and neck, pharyngeal arches derivative, or the conotruncal septum, these all are derived from the neural crest cells. So this is the summary of everything. What are the parts is developing from the in which part of the germ layer. So you have to memorize it, what the germ layers is giving to rise, this all everything. So this is everything about the 3rd week development or the general embryology. Now in the next section we are going to see the specific parts of our development here. So thank you, see you in next video, bye bye, best of luck.